thank you so much for having me and for changing things around. I also know I'm between you and dinner, so I'll keep it short and sweet. My name's Alexa. I am all right. As Cynthia mentioned, I'm at UMass Amherst in the Stockbridge School of Agriculture. I'm in Dr. Masood Hashemi's lab. And today I'm going to talk about one of our projects, garlic relay cropped into standing cover crops. So here's a photo of our lab. This is the Hashemi lab. Masood Hashemi is the team leader for the crop, dairy, livestock, equine extension team at UMass. Here we are in South Deerfield. We have a big research farm out there, and so that's where we conduct all of our research. And the research goals of our research group are to enhance the sustainability of farming by integrating cover crops into diversified cropping systems, to enhance nutrient recovery and nutrient cycling, suppress weeds, and improve general soil health. So we work in a variety of different systems and on a variety of different projects. So today I'm going to talk about just one of them. One of the reasons that we focus specifically on cover crops, as you can see, this is a field where there's no cover crops growing after harvest, is because it's a really easy way to keep plants in the ground at all times. And they're a really easy placeholder. Of course, they're not free. Cover crop seed costs money. But once you get cover crops in there, you can basically utilize free stuff from the atmosphere, so like carbon dioxide and dinitrogen from the air, to help get these resources into the ground. And so these living plants and these living roots are constantly feeding soil microbiology and expanding the soil food web, which is so important for soil health. And so here's a picture of a root tip. I'm not sure if you can see all the way back there, but you can see there's stuff coming out of it. All living roots produce exudates, which are basically carbon that comes out of the root tips. And that provides energy for soil microbes and for soil, basically expands the soil food web. And plants generally send out 30 to 40% of their photosynthates directly into the soil to feed soil microbes. And that's not because they're inefficient, that's because they're so reliant, and this relationship between microbes and plants is so important. So I'm sure you know about the 4 major principles of soil health, but this is something that we consider when we design our research projects. The first is minimizing soil disturbance. So of course, that's minimized tillage or reduced tillage or no tillage or perennial systems. Maximizing soil cover, again, perennial systems, or making sure that your soil is covered or planted at all times with cover crops. Maximizing continuous living roots. So again, cover crops are a great way to do that. And maximizing biodiversity. And so not to say cover crops do that again, but the main theme here is that 3 of these major principles of soil health can be addressed by planting cover crops, either by rotating or by providing mixtures, planting mixtures of cover crops. So these are some of the considerations that we think about when we figure out what research projects we're going to work on and what cover crops to plant. The first is anticipated goals. So when we're choosing a species of cover crops, why are we choosing that particular species? For example, if we have compacted soils, we might choose a biodrilling cover crop, such as forage radish or daikon. If we're looking to fix nitrogen, then we'll choose a legume like field pea or vetch. If we're hoping to recycle nutrients or build a lot of biomass in a short amount of time, then we might select something like oat. The second is production cycle or timing. So basically, when are you trying to have that cover crop in and out? When are you trying to plant your next crop? Sort of along those same lines, how are you going to terminate the cover crop? For winter kill cover crops, which I'm going to talk about today for this project, it's pretty easy because you plant them in the fall or late summer, and then in this climate, they will die in the winter. So you actually don't have to worry about terminating. They're going to die and then decompose in the spring. And then the last thing, which is really important, is residue decomposition and nutrient release and synchrony with your next crop. So just because a residue is decomposing and releasing nutrients doesn't mean that it's getting into your next crop. So one thing we work in our lab is trying out different systems to see if you plant a cover crop and you're expecting your next crop to take up, for instance, nitrogen from that crop, how much of that nitrogen might actually get into the cash crop that you're planting. So this is the project I'll be talking about today. It's mostly to do with garlic production in the Northeast. This is a project that was funded by the Massachusetts Department of Agricultural Resources, or MDAR. The project is designed to enhance the sustainability and resiliency of garlic production in the Northeast. The majority of vegetable producers in the Northeast grow garlic, and garlic is pretty unique because it has a very long and unique growing season. It's planted in October, and it starts to develop roots in the fall, but it doesn't sprout until the following spring in March, and then it continues to grow until harvest in July. So it's got a long growing season, and it's also a heavy feeder, so it requires a lot of fertilizer. Most importantly for our lab, it's a missed opportunity for cover cropping. So in the fall is a great time to plant cover crops, keep the soil covered until the following spring. If you're planting garlic, you're usually mulching with straw or planting directly into black plastic. So you're really missing an opportunity to keep a lot of roots in the soil and pick up a lot of biomass through cover crops. So we wanted to know if there was a way that we could build soil health and increase resilience without taking the land out of production. And so what we decided to try is planting cover crops the same way that we would in late summer or early fall, and then letting the cover crops grow and accumulate biomass all throughout the fall, and then directly planting garlic in between the rows of cover crops. And so for this project, we had small field plots. We did it by hand. We wanted to be very precise. Here's a basic planting pattern. You can see those little green grass looking things are oats or cover crops, and then the garlic's planted directly in between. And so we did it with a no-till dibbler. We tried to make furrows with the hoe, and then we also just dug little holes. We tried all different methods. We also wanted to see if we could scale this up, and so we actually tried to plant it with a no-till transplanter, which worked really well. This wasn't part of the actual research plot, but this was just something we wanted to try on the research farm to see if it would work, and it did, and we were happy with the results. So figured I'd show a video of that here. And so this is a cover crop mix. This is forage radish, field pea, and oat. And so we wanted to look at the entire system, and we were looking at a whole bunch of different things, including soil, food webs, nitrate. We also had a mycorrhizal treatment. We wanted to see if we could offset weed pressure with the system. We also wanted to see if we could offset nitrogen fertilization. So is there a way that we could actually fertilize less using the system? But some of the anticipated benefits that we were expecting to see are an increase in soil health and natural soil fertility, minimize soil erosion, and that's because not only do you have these residues on the surface, but you also have these living plants in the ground all throughout the fall, and roots do a much better job than passive mulch, such as black plastic or straw, at actually keeping the soil in place because they're using it and they're kind of just keeping it in place like a claw. They feed soil microbes both in the fall through the exudates, as we mentioned, and in the spring, because at that point the residues are decomposing. So as soon as the soil is warming out and the microbes are becoming active, you have these residues that are decomposing and starting to feed the microbes. Carbon sequestration and recycling of nutrients to growing garlic. So again, we're hoping for nitrogen to be recycled, but also other nutrients. If you're having plants continually in the soil, then nutrients are taken up into the plant biomass, and as those plants decompose, those nutrients are re-released. So you're hopefully keeping more nutrients in the soil and recycling from different soil depths and from basically utilizing all the nutrients that you have. Oh. Okay, so here's just okay, I went way too. Sorry. All right, so here's just a brief timeline. I don't know, it's a little bit cut off, but we planted cover crops in early September. The second year we actually planted in early August, and I'll show that in a minute. We allowed the cover crops to accumulate biomass throughout October. You can see we were hand planting in October. By November, they're starting to winter kill. By December, they're winter killed. And then March, you have the residues. April and June, the garlic is starting to sprout through the residues, and then we're harvesting in July. And one thing I'll say right away now, because I might forget to say later, I didn't include it as a slide, is that we did see a lot of weed pressure. So we did end up mulching. The first year we weeded all of the plots because we didn't want it to interfere with our results. And the second year we ended up mulching in the spring. So just something to consider. This is not effective for weed control. And so we chose 3 cover crops with different functions and various carbon to nitrogen ratios for this project: field pea, oat, and forage radish. We also planted a mixture of the 3. And so all 3 of these cover crops will winter kill. So they're not going to survive the winter. And so in the spring, we're not going to have to terminate them. So field pea is a legume. It'll fix atmospheric nitrogen. It's going to produce relatively high biomass, and it's got a low carbon to nitrogen ratio. So it's really leafy and hopefully will decompose quickly. Oat is fast growing, good at suppressing weeds, high carbon to nitrogen ratio. So it's a little bit woodier. And forage radish we chose because it was again low carbon to nitrogen ratio, but it was good at biodrilling, and also a lot of the biomass was in the soil. So it was a very different type of cover crop. And we're hoping we also chose the mixture because mixtures generally combine the benefits of various cover crops, and they also increase microbial biodiversity and above ground biodiversity. So this is just one thing I want to talk about before I get into the other results. So one of the first things we looked at is the biomass of the cover crops. And so you'll see in 2020, we planted early September, and in 2001, we planted 1 week earlier at the end of August. And this is not a huge difference, but I think we saw a huge difference in our results. And so I think that's really important because there's 2 ways that you can control the biomass of your cover crops. The first is seeding rate and the other is planting date. So the current recommendation is that for the fall, if you're going to plant fall cover crops, you get them in, especially annual fall cover crops, you get them in by September 15th. But everyone's busy, so it's really easy to push that back. But I think even in this scenario, you can see that there's such a dramatic difference, and just 1 week makes, if you plant 1 week earlier than end of August versus beginning of September, in some of these cases, you see 3 times as much biomass. So that's a huge difference. So timing is really, really important when planting cover crops. So here's just a visual difference, October 2020 versus October 2021. You can see there's a huge biomass difference already. So as I mentioned before, one of the goals of this project was to see whether or not we could offset nitrogen fertilization. And the graph on the right in that box, that's the nitrogen that's released from the cover crops during the garlic growing season. So the way that we actually track this is we make these little litter bags. When the cover crops winter kill, we harvest a bunch of the winter killed cover crops. We put them into these mesh bags made of window screen, and we leave them on the soil surface or bury them. In the case of roots, we actually separate the roots and the aerial parts because since they're winter killed, both are going to be there, and some of it's going to be underground and some of it's going to be on the surface. So we want to mimic that as closely as possible. And then in the spring, when the weather warms up and they start to decompose, every 2 weeks, we actually harvest those mesh bags and we look at the nitrogen content of the cover crops and the biomass. And so then we're able to make these curves. And so what we found is that during the growing season, there's not a lot of nitrogen released. So since garlic is a heavy feeder, 120 pounds of nitrogen per acre is what's recommended for garlic. We would not recommend offsetting fertilization with this. Interestingly, I actually have talked to a few farmers since we started this project, and one of them has been working with us who use this system. And there are people who use the system successfully and have never thought of it as a way to offset nitrogen, and they just fertilize the same way they normally would with great results. So, but that's just something to keep in mind. And with annual cover crops in general, that you might not see that great nitrogen contribution from annual cover crops, but you may see that nitrogen contribution from cover crops that overwinter if you're terminating them later on. So another thing we found, and this is that our garlic yield was actually reduced when intercropped with cover crops than when planted without. And even though that might look alarming, a few things to say about that. First, it's actually expected that you would see reduced yields from an intercropping system, because what you're doing is you're taking 2 different crops, and instead of planting them on 2 acres, you're planting them on 1 acre. So you're increasing your land use efficiency, but you may see decreased yields from both crops. The other thing to consider is that this is a transitional system. So we planted this in a field that has not been cover crop before and has been excessively tilled. And so, as I mentioned, I've been talking to other farmers, and I know one farmer who's been doing this for many, many years on a no-till system on a large scale. And he has really, really great yields and doesn't have this yield lag. So this might be something to do with just the transition period. One way that we can offset this, I think, is playing with seeding rate. So the first year we had, as you can probably remember, that really low biomass of cover crops, and we didn't see such a big difference, a dramatic difference in yield, versus the second year where we had a lot of biomass with the cover crops and we saw a more dramatic yield difference. So playing with seeding rate may actually offset this. The other thing I should mention that we did that is different than maybe a lot of garlic growers is that the recommended nitrogen application is you start fertilizing in the fall. So you make your first application when you plant, and that's because that's when the garlic is first starting to develop roots. And then you make your next 2 applications as split applications in the spring. And we didn't apply nitrogen in the fall. And the reason we didn't is because we didn't know where that nitrogen would go. We also have the cover crops there and they could take that nitrogen up. So we didn't want to basically confound our results. But it may help to apply that nitrogen in the fall because that's when that garlic is getting established. And so giving it that extra boost might actually help offset these yield differences as well. The last thing is that it actually wasn't our goal to increase garlic yields. I mean, we know how to get these really high garlic yields already. Our goal was to increase soil health and resilience. And so and when we say resilience, that means basically being able to produce yields in the long term. So how can we make our soil resilient so that it can withstand drought or heavy rainfall and continue to produce? And so this is a way that we can actually integrate these practices of soil health without taking land out of production. And so just kind of a last slide, these are some of the other projects our lab was working on. So if you're interested in any of these, we mostly don't work on vegetables. It's primarily a forage lab. So if you're interested in any of these other projects, feel free to talk to me and I can connect you with the right people. Or of course, if you're interested in any of the other data that I took or more about this project, feel free to email me or talk to me right now, and I will be happy to answer questions. Yeah, so cloves, no, which we did measure, and I, in retrospect, thought didn't make sense too. But diameter, yeah, there was a difference in diameter with the higher yielding. Higher yields were usually higher diameter. Interestingly, I didn't present these results because we had 15 minutes and they were kind of more complicated. But yes, there was a difference in the quality. We actually measured the nutrient content of the bulbs. And so we measured phosphorus, potassium, copper, manganese, just several nutrients. And there was a difference in the different years among the different cover crop treatments. So one thing I didn't mention and I should have is that we planted these plots in the same exact location both years. So this was the same field. And so like, for instance, in the second year, we saw much higher phosphorus uptake in the bulbs in the oat plots as compared to the other plots. In the first year, winter pea had higher phosphorus uptake. And so there were these small differences that we did see in the quality, which I think is important to also consider when you're using cover crops, is that because different cover crops may uptake nutrients at different rates and in different amounts, and they're going to release them differently. And so that will impact the quality of your crops, which was cool. I mean, just looking at it from the nutrient density perspective, actually seeing these differences. So we didn't actually measure the organic matter level, so I can't say definitively, but one thing we are looking at, and I don't have the results for this yet, is we're looking at the soil food web, and so like actual microbial activity in the soil. And so hopefully what I'm hoping that we see is that we see differences in like the actual food web structure, and which is not organic matter, but that's one way that we'll be able to see if there were differences after the 2 years. But realistically, one thing I will say is that we did look at just microbial respiration in both of the years. And we didn't see differences in the first 2 years, which isn't that unusual too, because some of these systems, I think, they take a long time. It's like cumulative kind of, you're looking at a cumulative results. So 2 years might not be enough to see differences in, for example, microbial respiration, but that doesn't mean that there's nothing happening. It might mean that you might have to wait 3 or 4 or 5 years to actually see those differences in like a lab, We did buckwheat. So that was easy because it was quick and it was easy to terminate in time for us to plant cover crops again in late summer, early fall.